am calling to order the 1244th regular meeting of the Entomological Society of Washington. And as I stated earlier, please, if you're in person and eat, contribute to the Venmo or the cash. The voice order of business is Secretary Gary Hebel reading the minutes from the last meeting.
On March the 7th, 2024, at approximately 7 p.m., the 1243rd regular meeting of the Entomological Society of Washington was called to order by President Don Weber in room WG 33 in the National Museum of Natural History. Present for the meeting were 14 members, 6 guests, and 20 virtual attendees. Recording Secretary Gary Hebel read the minutes of the February meeting, and these were approved by the audience. President Weber noted that President-elect Andy Jensen is planning on utilizing the log lodge in Beltsville for the annual meeting, and that the event is scheduled for June the 8th. The speaker will be Hugh Douglas. Membership officer Elizabeth Young announced that she has secured a position with aphids at the museum. President Weber noted a new book, Metamorphosis, which will be printed in a few weeks. There is also a new book, Darwin for Beginners. Past President Matt Buffington noted progress in a procedure to determine fire ants. President Weber noted that the ESA is welcoming comments for current investigations on name changes for 9 insect species, including Asian longhorn beetles and Asian rice borer. Matt Buffington offered comments on the recent death of David Smith, who was 87 and a pillar of the Hymenoptera community. Dave had run malaise traps for many years, and resulting collections strongly contributed to research studies by colleagues. He was an active researcher of sawflies, including worldwide species, and contributed 407 publications to Science. Jonah Ulmer worked with Dave during the year, first by emails and then in person, and commented on his strong enthusiasm for taxonomic work. Another member of ESW, Ralph Eckerlin, has also died recently. Ray Gagne had clipped an obituary from the Washington Post and requested Gary Hebel to read highlights from it. The obit revealed a life devoted to science with a passion for parasitology. He was the co-author of an exhaustive compendium, The Syphonoptera of North America, soon to be published in the Annals of the Carnegie Museum of Natural History. Program Chair Alan Norbaum introduced the speaker of the evening, Luke Pressley, whose presentation was titled Mandibular Rods and Parasitic Wasps, the form and function of a novel synapomorphy for the Hymenopteran infraorder Dr. Dr. Presley first offered a brief history of how he became interested in the spatial subject of mandibular rods. In 2017, he worked with John Harity, who introduced him to the subject. Another colleague, Gary Gibson, couldn't help with the subject, commenting, "Don't ask me." In 1919, mandibular rods were recognized as structures that appear from the apex to the end of mandibles. The rods are absent in certain groups. It has been recognized that there are 2 choices of ancestral development. Ancestral state reconstruction was chosen for study as a likely developmental plan. John Harity said that use of molecular data results were not demonstrably true. In a study by Vanderkamp in 2021, it was demonstrated that some teeth don't have rods and that rods are hollow. Recent analyses indicate that much more work is needed for this research area. The meeting was adjourned at 8:20 p.m.
Are there any corrections, additions, deletions to the minutes? Thank you, Gary. I don't know if that Darwin for Beginners was a new book.
I'm not sure either.
Yeah.
I was wondering that myself. I seem to remember it being.
It wasn't
Yeah.
new.
That was
it
an
just kept-
interesting book.
Yeah.
Yeah.
That'll present. I was hoping you'd present, and Matt would be very happy to hear this. He's escorting a few late members in. I was hoping to present a draft of the revised bylaws, but there was a lawyer. So, that might take a little bit. But I do want to thank Abby Kula, who's probably online. Yes. For all her work on this and several other recent treasurer duties. You notice, is Andrew online?
Abby's on.
Andrew Jansen. I think Gary summarized that we're gonna have the banquet Saturday, June 8th at the Log Lodge, and we are gonna arrange for some shuttles from the Greenbelt Metro. And who is the guest from Ottawa? Gary.
Hugh
Douglas.
Hugh Douglas.
Hugh Douglas. I believe.
Yeah. All right, that's all I have on that. And also, we're working on revising the proceedings of the Ensac of Washington cover, which I think Jerry's taking the lead on with Mark and the Pubs Committee. It's somewhat out of date, and there is a United States Postal Service regulation that says they have to see the 2 covers of the most recent journal to grant a mailing permit. Go figure. All right. Welcome. So also, Toledo, do you have any updates on the publications, hard copies, or how's that go?
I don't have an update on the amount we have already, but we just received the archive copies for volume 225, number 3.
Okay. And so do you need help with the hard copies still, organizing?
Yeah, I need help counting it, but I first need to figure out when I can do it, to then send an email to everybody asking people to help me.
Okay. Pending that. And Lourdes is not here. I think she was gonna bring.
She's on.
Oh, but she can't bring the bling. Lourdes. so we-
Hi.
Yeah,
So we did get the mug. And it looks really good. We put it through the
dishwasher.
dishwasher. But it is taking some time to get the shirts. So they're probably different distributors, and it's been almost a month. But it's similar to other societies that I've ordered things from.
You
So,
mean that kind of trickles in?
Yeah, and it takes a while. But if Matt's there, he can tell about the quality of the mug. We thought it was really good.
The mug holds coffee. Yep.
That's the basic
And it washes.
function of a
It's
mug.
good you didn't belabor
Yeah,
that. Thank
the
you.
logo, in my opinion, is too big and it's in the wrong location. But
Fair enough.
it's actually decent quality. So, and it came really fast.
Yes, so we'll adjust the logo to, because it's sort of in the front, it's not on the sides, sort of, if you know what I mean. I can run and get it and do it for show and tell, and turn on my video. By the way, your video is off. I don't know if you knew that.
Oh.
Oh.
I think it's that. In other words, you're saying you can't see the speaker.
Right.
That's okay.
Was it?
As long as it's just you.
I can see Al.
Al's working on it.
I thought it was a problem with my camera, but yeah, or my Zoom, but yeah, that's true.
In the meantime, do we have new members and visitors?
Hello, hi there. I don't know if I can turn on my camera. Hello. I don't know if I'm recording. Hi, it's Elizabeth Young. I'm the membership secretary. We've got 4 new members to announce this month. Their names are Jeremy Sloan, Lance Jones. Sorry, that's my dog. Sorry, and actually 3 new members, not 4. Sorry, and Yavuz Turan. And if there are any new members or visitors, oh, I'm so sorry. If there are any, oh gosh, if there are any visitors who are here, I'm so sorry. If there are any visitors who are here today at the museum or on Zoom, please announce yourselves. Thank you.
All right.
I'd like to announce.
There are no objections to these members joining the society. It's so ordered. Let's see. Do we some guests?
It's my husband Owen.
No, I never got that.
All right, Laura Bonfor announced. Or, anyone else? Yeah.
To introduce Jess Awad, she's visiting us from Germany for the next week and a half,
All right. What's she studying?
working on platygastra and wasps.
All right, that makes sense. Okay, welcome. Anyone else? And you're looking for what?
I think the camera's just off.
Okay. Mavens of IT have arrived.
There we go. There we
Um,
go.
Yay! Oh, I'm so happy to see you all. Hello. This is my dog Tetra. She wanted to
Now,
say hi.
now we can see everybody.
Well, thank you so much for coming.
yeah,
Are
it's
there multiple?
it's that camera.
How do you change it? So what do people in Zoom see? They see us.
They see that.
Yeah.
That's what they see.
All right. Well,
No,
All right.
switch somewhere.
I'm thinking.
Yeah, it's not necessarily that important. All right, do we have any unfinished business?
Getting the camera.
Yeah, the unfinished business is getting the camera to work. But
Is it literally just we need to turn it?
no, you can't take that one. Any new business? Any show and tell? That is presentation of notes and exhibition of specimens. Anyone? Come on, okay.
I have a thing that I showed off earlier today, the event at the Smithsonian, but I still have it, so I was like, why not do it again? Oh, hey, Brent. So these are 3D printed models that we printed out at the USDA facility in Beltsville. If you came to the last ESW talk, I did a talk on mandibular rods and I printed those out because they're what I'm all about. But it's this like nice, really robust carbon fiber plastic. And they came out with really high detail. So highly recommend.
How many X is that?
Many.
Yeah, come on, 500X?
I think probably close to it. They're compared to what I should have brought the wasp too.
Oh, you should.
Next time.
Next time. Yeah, we'll
Could've
keep
been
this
a
whole
good
thing
time.
going with mandibular rods. Every week I'm gonna bring it back.
All right.
Yeah,
You said it's in there as well. We wouldn't. No.
It is neat and it's fun.
Okay.
I guess we're gonna look at the audience. Oh, yes. Lourdes, did you have something? You raised your hand.
Saw the mug.
Oh, the mug.
Yes, I located the mug.
Yeah, Sarah. Can we-
So
oh,
if you
like-
see, it's, if you hold
Lord,
it, logo
Jerry.
is sort of.
Well, she's showing.
No, she's showing.
If you
Can you
write,
see?
Can you
Can you see?
like
Sorry.
click this and it does it? No.
hit the dots.
Yeah.
Okay, I'm not showing yet.
There it is.
Oh, we got one.
All
Yeah.
right.
So it's there, but if you hold it like this, so the logo is gonna be here.
Scientific
and
name
here.
been shown.
No.
I don't know.
I sure
No,
don't.
It's micro deleted.
Yeah, Good, you guys can see it?
Yeah.
Very
So it's a little large.
I drink a lot of coffee.
No,
No,
no.
it's not.
Logo.
Oh,
No,
I
it's not that, not that big.
Oh, it's blue inside.
It's too big and
Charming.
Oh.
All right, now how do I send it back?
There, okay, thank you.
Blue pen,
Yep.
like that.
All right.
Yeah, we tried to get the inside cornflower blue.
Yeah, really?
The cornflower
I
blue.
don't know, it didn't work. How does one get a mug? That's pending. I think it's pending a remake of that mug that shrinks the logo of that, right?
Well,
And-
yeah, and I think we're gonna, make it available so people just pick it from the site.
Right. It's gonna go on the site probably, I would guess, by the May meeting. That's not a promise, but Lourdes will take care of it. She's very good at that stuff.
Great,
All right.
thank-
Any other exhibition, specimens or notes or books? Okay, has everybody signed the clipboard there? Well, now we come to what you all have been waiting for, and Alan will, what? Did I skip something?
Aphids.
A, okay, aphids, not aphids.
No.
Yeah. Alan, you want to introduce the speaker?
So Gary, we have 15.
Somehow we have.
I'll introduce the
I did
speaker.
on
Next month in May, the speaker will be Karen Burkhart from the University of College Park. Now I have the pleasure to introduce Becca Clement. She completed a BS in biology at Brigham Young in 2017, where she worked with Seth Bybee and Riley Nelson and studied Diptera and Proteonaza. She completed a PhD in 2022 at the Computational Biology Institute at George Washington University, working with Keith Crandall and Amy Zane. Studying termites and microbiomes. She's currently the USDA Scientific Computing Initiative postdoctoral fellow at the Systematic Entomology. Her research interests are using DNA and computational tools to understand the evolution and ecology. And she's also working on her insect songbirds. Suggest that's something good for the banquet.
Yeah, bring it back.
And her presentation tonight is hybrid superbugs. One way aphids are taking control.
That's my mom.
Aw.
Mom, mute your thing.
Somehow.
Okay, awesome. I'm very excited to be here with you guys talking about aphids tonight. And if we have time, then I'll also be sharing some of my insect songs with you. So it's a work in progress. If you were at the Smithsonian thing back in December, I have one more song. So or it's sort of like half a song. It's sort of work in progress. But yeah, today I'm going to be talking about aphids. Like I said, I work with Dr. Chris Owen on the aphid team, and this is us in New Mexico last summer. We had a lot of fun going there. But my background is, do you think we can hide these things?
There's a way to hide it.
Try and press escape, maybe?
There's a way.
There's a full hide video.
Go in there.
No, you had it.
More hide in your panel,
Hide you.
and then more hide for the main controls.
Wow, okay, great.
Hey.
Yeah, awesome. So I am what I consider myself a taxon shifter. I during my undergraduate, I worked on dragonflies and flies, and then during grad school, there was a chance to go to Australia, and so I worked on termites. But I love insects, and I love trying to understand the way that things work. Where insects are, why there's insect diversity, and what's going on. During my PhD, I focused on Australian termite diversities, a little bit of their about their role as decomposers and their biogeography and their microbiome as well. But I also didn't just work on insects because I was in a computational lab. So we worked on things like human microbiome, cow microbiome, and even during the pandemic, I worked on a COVID project. So I love being able to use DNA to answer really cool questions. So this figure sums up all of my research. So I am really interested in how do things like environment, evolution, microbiome, species interactions, human interactions, impact species distributions. And then what are the downstream consequences for dispersal, disease, and decay? And invasive species actually kind of encompasses all of this. So if you bring up aphids here, you can kind of see what a cool system this is to work on. Aphids are very agriculturally important, and so their presence is affected by human interactions. Is there a way I can make the arrow bar? Okay, no. Yeah, anyway. So we've got aphids, the system that's definitely influenced by humans because humans are bringing shipping vegetables all over the place. And you definitely have some awesome species interactions going on here. So not just between the aphid and the plant, but between the aphids and the parasitoids that are trying to kill the aphids. You've also got, one of the reasons aphids are important agricultural pests is because of they carry plant viruses. And so they transmit a plant virus from plant to plant, which is really bad for us and bad for the plants. They also have really great symbiotic relationships with their microbiome. You might have heard of Buchnera, which is a type of bacteria that because aphids are sucking out sap from plants, they're not able to get all the nutrients that they need. So they actually have this symbiotic relationship with Buchnera, which allows them to be healthy. They also produce honeydew, which is like a sweet substance. And because of this, they have a relationship with the ants. So the ants sort of keep them as pets. And as a result, it's actually great for the aphid to be a pet because the ants will chase off the parasites. So lots of really cool interactions going on, and it's really fun to be working on this system. Aphids are also the most invasive, the most species-rich group of invasive species. So there's 430 invasive species, which is more than any other insect group. And in fact, if you were to go outside right now and find aphids on plants, about 30% of the aphids that you find are going to be non-native aphids. So they could be from somewhere really far away. I talked about they cause billions of dollars of damage. A lot of this is from mechanical damage. One aphid can't really ruin your plant, but if you have 1,000 all in the same leaf, it's definitely going to suck the sap right out of your leaf. They pass plant viruses, and the honeydew, although it's really nice for the ants, it can make mold grow on the plants, and it's also can gum up the cotton machines. Anyway, this figure shows these are the 45 major insect pests for the 6 main food crops, and you can see like aphid have their own category within Miptera. These are kind of bad. So what makes aphids so good at being invasive? First of all, they reproduce quickly. Here's a picture of aphids that have been really successful. Part of this reproduction is asexual. So aphids have a unique reproductive strategy. They have telescoping generation. So here you can see this is a mama aphid, and she is currently pregnant. But these babies are actually also, when they're born pregnant. So you've got, a lot of this makes it really, really easy for aphids to like get a lot of generations in a short time. However, you would think, but they're going to lose out on the diversity that comes from sexual relationships. Well, aphids have the best of both worlds. So most of the year they are reproducing asexually, but then during the fall, they go off onto usually a different host plant. And then they produce males, and then they reproduce sexually, and then they lay eggs that can last over winter. So anyway, they're amazing at what they do. There's also a lot of aphids that are generalists. So for the most part, most aphids are restricted to a single host plant. And a lot of them are also host alternating. So most of the year they'll be on one plant, and then in the fall, they go to the other plant to reproduce. However, there are a few aphids, like Aphis cassipii, that can feed on 135 different plant families. So this figure shows the 66 most prolificous aphid species. And this is the number of plant families that they have as hosts. And so 3 of these up here, Aphis cassipii, we are talking about Aphis today. So Aphis cassipii, Mysis, and Aphis bobbie, these actually have up to 135 different plant families that they can live on. So this makes them really good at being invasive. Okay, so they're good at reproducing. They are, a lot of them can be prolificous. And then finally, they're also tiny. So because they're tiny, they can be easily transported on your vegetables. And we do a lot to bring them into different places. And because of this, it gives a lot of opportunities for hybridization. So you think about one of the things that makes me amazed by invasive species is that you think usually if a species is adapted for one type of environment, it should not really beat out all the competition in the other environment. However, in a lot of cases, we see invasive species outcompeting the natives. And so one reason that this could be happening is because of hybridization. So this is sort of like a flow chart of what we think sort of happens when aphids are hybridizing. So basically millions of years of evolution go by and you have aphids that are living all over the world. And then one day we bring some lettuce over on a boat to a different place. And then all of a sudden these 2 aphids that previously were separated, all of a sudden are able to reproduce and hybridize. So how does it happen? So hybridization is just interbreeding of individuals from 2 different populations or groups of populations, and there has to be a hybrid zone. This can be a geographic hybrid zone, but this can also happen because of a shared host plant or it's a time. And why does it matter if it's happening? Well, first of all, it can speed up evolution, and it can also lead to new species. So in invasive species that we don't really like, this can lead to things like temperature tolerance. So especially as we have climate changing, you have species that are better able to survive hotter temperatures. This can lead to genetic variation. Sometimes hybrids are more able to evade predators, and sometimes they are have more pesticide resistance. So there have been a lot of different examples of invasive species where they actually have found hybridization, and there has been some initial things pointing to maybe hybridization going on in aphids. So this is what prompted me to be working on hybridization in aphids. So the question that I'm asking, first of all, is do we find evidence of hybridization in agriculturally important aphid genera? And then second, is this hybridization more prevalent in prolificous aphids? Because they've got that hybrid zone, they're able to share plants with more aphid species. A little bit of background of aphid systematics. This is the Hemiptera tree. Aphids are true bugs. They're in the Sternorhyncha, which includes the scale insects, the psyllids, and the whiteflies. And so there's about 5,000 species of aphids in total. And the groups that I'll be focusing on today are in the subfamily Aphidaini in 2 different tribes. So first of all, we've got the genus Mysis, which is right here, and the genus Aphis, which is right here. So they're in 2 different tribes, but they're both agriculturally important. First of all, we have Aphis. There's about 600 species of Aphis. They're mostly northern hemisphere, and there are a bunch of different prolificous aphids, including soybean aphid, cotton aphid, citrus aphid. In Mysis, there are fewer species. There's only about 55 species, but they've got a cosmopolitan distribution. And again, a lot of them are really prolific, including this Mysis persicid peach potato aphid, which has over 400 species. So for this project, first thing I did was I generated trees. Second of all, I generated SNPs, and then finally I tested for hybridization. So first of all, the data that I used, it was shotgun sequencing data. We had 31 aphid samples that we generated and 21 sequences that I got from publicly available reads. About 76 million reads per sample. And again, this is shotgun sequencing. So you take the whole genome, you take all of the DNA that's there, you chop it up into really tiny pieces, and then later you come back and assemble it into contigs or hopefully genomes. So, okay, so the first thing I did for this project, when you're making phylogenies, it's really important that you have orthologs. And so I took known genomes and for both Aphis and Mysis, and I found some single copy orthologs. Yeah, because you don't want to be using, we know that gene duplication happens. It's really important to have these single copy orthologs. And so for Aphis, there was more than 8,000, and for Mysis, there were about 7,000 single copy orthologs. So then I built, then I used sequence capture to take these 7,000 reads, these 7,000 genes, and then I took all my shotgun sequences and mapped them all to these reads so that I came out with about 7,000 genes, or we call them genes, but they're really orthologs. So, and then finally I built some trees. I built a mitochondrial tree, concatenation tree, and the gene tree, species tree. So this is the first, here's Aphis tree. We've got the red stars indicate the most prolific species in the group. So you can see there are prolific species all throughout the tree here. And here's the concatenation tree. And here is the Astral species tree. And they have almost identical topologies. And again, we're using 7,000 taxa, so these are really highly supported. I think this node right here is the only one that didn't have 100% support. The mitochondria tree was also very similar, except for these mysterious taxa that are labeled a orange triangle, which I'll talk about later. Then here's the Mysis tree, same story. We've got a very similar concatenation tree as the species tree. Again, there are a few really prolific species here, and the mitochondria tree has a similar topology. We'll talk about the triangle later. Okay, so I got my trees. This is great. This is actually really important because even though these are really important invasive species agriculturally, they're not good phylogenies for them. So next of all, I generated SNPs. So a SNP is also referred, a SNP is short for a single nucleotide polymorphism. So when you make DNA alignments, there will be some sites where you'll actually have different, most of the alignment will hopefully be the same. But you have little sections of your alignment that might be one base pair different, and these are called SNPs. So we took all these SNPs into a file, we put them into a VCF file, and then I filtered based on the quality and coverage, and then I ended up with about 585,000 SNPs for Aphis and 1.2 million SNPs for Mysis. Again, lots of data, that's great. Finally, we get to the point that we can test for hybridization. So as I talked about before, you can imagine, here's this figure, I showed it a little bit before, but you can imagine this is what hybridization looks like, right? So here's your timeline. Eventually you have a speciation event happening here. However, right here, you've got this hybridization event, and all of a sudden the rest of these species have some sort of trace from the other species because of this introgression or hybridization event. But it's really tricky. It can be kind of tricky to tell, is this happening because of hybridization or is this happening because of incomplete lineage sorting? So this figure, I think it does a good job explaining why we might see our gene trees be different from our species trees. So you can imagine a life where you have, here's a species, here's your ancestral population, and the outgroup is very similar to it. then you've got a speciation event here, right? And we've got this species, species 1, goes over here, and then it's got a bottleneck event, and eventually you've got species 1 has the number 2 version of this allele, the number 2 version of this allele, and the number 2 version of this gene. This is gene. There's 3 genes in this imaginary species, and each of them has 2 alleles, right? So this one's got alleles 2, 2, and 1. Then over here in the other species, you've got it breaks off, and one species, this becomes species 2, and this becomes species 3. So you imagine if we're thinking about phylogenies and how things are related to each other, you should expect to see your species tree looking like this, right? You've got your outgroup, and then 2 and 3 are more closely related to each other than 1, right? However, if you actually look at the individual alleles, you'll see here that species 3 and species 2 have different alleles. This guy has 1 and this guy has 2, right? Whereas this one over here, species 1, actually shares an allele with species 2. So you'll actually get your gene tree will end up looking like this, showing you that 1 and 2 are more closely related to each other than they are to 3, right? So this can happen a lot, and we talk about this a lot in phylogenetics. But usually because it's something that sort of happens randomly, you expect that 50% of the time you're going to have your gene trees looking like in one topology. Most of the time you're hoping that your gene tree looks like the species tree, but 50% of the time you're going to have it looking like one of the alternate trees, and 50% of the time you're going to have it looking like the other topology. However, if there is hybridization happening, and say these two were actually hybridizing, they somehow made it across the river, then you would expect to see many more of the trees that look like ones that have 1 and 3 together, right? So that would be like this. You'd expect to see way more. So you can actually look at the percentage of trees that have, if there are trees with more, one topology has more trees than the other alternate topology, then that's often a sign of hybridization. So we call this, with SNPs, we call this the ABBA-BABA test. So basically, you can either have your tree looking like an ABBA formation or a BABA formation. And so I used a software called D-Suite with my SNPs that I generated, and I ended up with, and it tells you it'll give you a D score, and if your D score is high enough, then you know, oh, this is more or less than 50%. So, so here's the results from D-Suite. It found that there was admixture between a bunch of different species of aphis, and of note, Aphis cassipii and Aphis glycines are both polyphagous species. And in Mysis, we also found admixture between Mysis varians and Mysis antirrhini. Mysis antirrhini is really polyphagous, and Mysis varians is a really important test of plum trees. So we can do this using the SNP values, but you can also do this using the trees. So I generated a lot of gene trees already, and then I used a software called SNAC, which allows you to basically do the ABBA-BABA test, but with trees instead of with just SNPs. So I did it, and this told me that there were at least 2 hybridization events happening in our aphis tree, and also 2 hybridization events happening in my aphis tree. Another way to view this is a publication from Edelman et al. 2019. They found hybridization in Heliconius butterflies. Another way to look at this is by overlaying your gene trees on top of your species tree. And here they did, and they found, this was their interpretation. They said, okay, so it looks like there's a hybridization event happening here, hybridization event happening here, and a hybridization event happening in a bunch of different places. So I did this with my trees. This is all of my species trees. This is all of my gene trees mapped onto a single species tree, or I guess the species tree is not exactly here. But you can see in these aphis species over here, there's a ton of introgression happening. So these trees, there's not a single topology. That means there's a lot of interbreeding happening between these aphis species. Mysis, it looks like there's a little bit less. There are more concrete trees that you can actually see, which says that maybe there's not as much. Mysis is also like a bit of an older lineage than Aphis is. So, and then we finally come to these orange triangles. So, if you notice here, these are kind of outgroups. These two specimens were both identified as Mysis. However, in our phylogeny, in the mitochondria tree, both of these are, if you use just the mitochondria data, they seem like they're Aphis. So originally I thought we might have some like crazy wild hybridization going on. Anyway, I talked about this last time, and most people I've talked to about this say there's no way that that can happen. You can't have multiple tribes. It's probably contamination, right? So what I'm going to show you next is fresh off the press, looking for contamination in your sequences. So I already encountered this problem once in my tree. So we've got parasitoids in our aphid species, and sometimes you try and sequence an aphid, and you can't just, like, if some insects are big enough that you can just sequence a leg, aphids, like, you got to do the whole thing. You got to do multiples, or else you're not going to get enough DNA because they're so tiny, right? But this species right here, these two in green are supposed to be the same species, but because one of them had a parasitoid in it, it actually came out as like a sister to the whole rest of the aphid tree, which is the problem if you care about topologies here, right? So enter blob tools. I'm very excited to be able to share this to you. This tool is very cool. If you want to use it, talk to me because I made a lot of mistakes, and if you don't, then I'll. Yeah, it's great. It's great. So basically, what you do, the idea behind this is that you can tell if a sequence belongs to. But I think it was originally built for like detecting if there's differences in bacteria. You can tell. So you have your sequences, you put them into contigs, and then you take these contigs and you blast all of them, see if they match anything on the NCBI database, and then you look at how much coverage they have. So you take all your raw reads and then you map them back to your contigs, and you see which contigs have a ton of them and which contigs have only a few of them. For example, if you are looking at mitochondria sequences, you should see that there are like a really high coverage of mitochondria. Because if you're just chopping up and sequencing all the DNA, like a lot of it is going to be mitochondria, right? But this actually separates out. Okay, yeah. So you've got on your y-axis here, you have the coverage. So how many times did your reads match, right? And on the x-axis here, you have your GC content, because some species have different GC contents, right? So aphids should have a different GC content than other species. So this is Aphis cricivora. This is one of our species of aphids. This is like a normal one. And here you can see, so the light blue, I think the size of the circle is how long the contig is, but the light blue shows all the arthropod sequences. The dark blue didn't match to anything on GenBank, and the light green matched to the phylum Pseudomonodota, which is Buchnera. So we've got like we're bringing back, and we see that all of those all of these. Yeah, so these light green ones up here are all Buchnera. There's also some other cool stuff in here. We've got orange is Wolbachia. And Wolbachia is a common symbiont of a lot of insects, but looks like it's here in this aphid too. And then there's a few other types of bacteria here as well. I will say the limitation of using this is really your reference database, right? So I'm looking for Aphis coreopsidus here. And so this is just looking at all the arthropod data. And most of it is matching to Aphis at the genus level. but it's really matching to Aphis specifii, which is the cotton aphid, which is our most commonly sequenced organism of the aphids, right? So it matched to a few other species of, and it matched to a few other genera of aphids as well, but it's mostly all aphids here. So what does it look like when we have a known contaminant? So here's our Aphis aurantii species specimen that I know had a parasitoid because it came out in a weird spot in the tree, right? And so you can already see that there are different blobs. Right here we've got green again, is the bacteria. Looks like dark green is some plants. We may have even gotten some chordata. So you know, I actually looked and it was I was thinking it was probably like human because we were like as we were scrubbing stuff off, I was like, okay, but it was like the thing that it mapped, the genus that it mapped to is like the hippopotamus. I don't think I think it might just, I don't know exactly what it is, but I don't know exactly what it is, but you can only trust this data so far because if it's not in your reference database. I bet it was some sort of bacteria that maybe was, I don't know. So here's what it looks like on the phylum level, but here we go to the order level. So we've got Hemiptera, which is what the aphids are in green, and all this blue up here is Hymenoptera. Isn't that cool? So now I can actually sort out these reads, and I can take I can say, okay, don't like get away anything Hymenoptera. I don't want to look at that. Just give me things that have coverage of like 15 or less. And then all of a sudden this data that before I had to like just throw out of my tree because I couldn't use it, but now I can use it because I've sorted it by its coverage. It turns out this, when we look at it at the species level, most of our DNA is mapping to Aphidius gifuensis, which is this little parasitoid wasp here. Here you can see one that's crawling out of an aphid mummy, so it's wild. It looks like we've also got some Zila albiditarsis, and all of this is like, I don't really know if there are really two different parasitoids that had parasitized this aphid, or if it's just like some of the reads are mapping to the NCBI database. And it might not be Aphidius gifuensis either. That's probably just the most common thing on NCBI. Anyway, so can this method detect contamination in my question-causing samples? Short story, no. So we've got, there is something weird going on here. So here's arthropods. Again, the light green is the bacteria. But when I map it to the genus level, it's not mapping, it's actually mapping to a Surthosiphon, which is a different genera. It's neither Aphis nor Mysis. So I don't really know what's going on in this situation. Again, this is fresh off the press analysis, but it's fun. But anyways, back to our question, do we find evidence of hybridization in polyphagous pest aphid species? Yes, we do. Is there this intergeneric, intertribal hybridization that's going on? It's really unclear. At first I was kind of like discouraged by this because I would just like to know what is happening and really what is going on. And I talked to some of the people who work on the hybridization software at the University College of London. And they told me like, it's okay to be discouraged about this right now because like, we're still learning a lot about hybridization. Like it's a pretty new field and we don't really know what to do with this. And we don't know, like, do we really see how much hybridization can happen and what does it do to our biology? So anyway, there's, I think there's going to be a lot of stuff coming out. I think there is already a bunch of people finding hybridization. And the next step is like really wondering what we can do with this. I think in a USDA position, it's really important to be aware that this is happening though, because it can lead to morphological and genetic anomalies in identification. And then the follow-up question to this is like, what effect is it going to have on resistance or invasion? So, in conclusion, life is an enigma. It's more complicated than a bifurcating tree. But DNA can give us insight into insects. And again, yeah, hybridization research is new. I'm sure there's going to be I'm sure we're gonna keep learning and figure out how much of a role this is actually playing in insect evolution. But either way, invasive species can cause real problems, and hybridization can be a secret to their success. This figure over here is during my PhD. These are not aphids, these are termites. These are 12 different termite species that I collected in Australia, and I worked with the artist. And because of my work with the artist, I feel like she's sort of inspired me, and what you hear after this is going to be the thanks to inspiration from Donna on how you can bring science and art together. So I'm also working on DNA data is one type of big data, but there's a bunch of other big data. So part of my postdoc was organizing a biological invasions and big data working group where we're looking at how to use like occurrence data, social media data, genomics data, AI, machine learning to address invasive species. And that should be coming out soon. So, I mean, not coming out soon. That's almost, you can submit it soon. Anyway, big thank you to all of you. And if you have questions, I'd be happy to answer them.
Your guitar?
Oh yeah, if there's no questions, I can.
I got a question.
Okay, what's your question?
It's a general question. So if you've got a good talk, by the way,
Yeah.
great talk, but you've got, so this relation, no buts, you've got this relationship potentially between polyphagy and the degree to which things may hybridize, or the degree to which things may be distantly related and still hybridize. It seems like between the cloud diagrams and the blob test, then, or the blob tools, it might be possible to almost quantify that, because you're basically just asking whether the extent to which the ability to interbreed is plesiomorphic, right? I mean, the further down the tree they can interbreed, and if you can identify hybridization events at different nodes, you could potentially quantify that on the basis of tree structure. In other words, if you can identify the hybridization events, you can identify, like Gossipii is hybridizing with everything. And
Right.
if that's not just an artifact of how commonly it's collected, if it's actually related to polyphagy and how frequently it encounters other things, blah, 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 then, and you can identify those nodes, would it be possible to start grading hybridization assessments, or, coming up with tiers of hybridization, whatever?
I think it is kind of difficult to know. One thing I'm kind of struggling with is, yeah, we've got really invasive ones, we've got really prolificous ones. How do we peel apart? Are you saying you know how?
No, I'm asking. I'm not playing
Yeah.
20 questions.
It does
Trust
seem
me.
like you should be able to.
Yeah.
I haven't quite figured out
It's
how.
just interesting alone.
Yeah. Don't-
So when you're trying to parse out the parasitoids from the aphid, how deep do you go with references? Do you count for like hyperparasitoids, like Dendroceras or aphids, which both parasitize the conids that are parasitizing the aphids? So
Well, in theory, you should be able to get all of the DNA. So as long as there's enough DNA that it's showing up, and here I've got like 70 million reads total. So I think it would pull out. I think the bigger issue is there going to be reference sequences on NCBI that you could actually match to?
Yeah, so if you're pulling out everything, right, and just throwing it into and blasting it all, so if
Yeah.
it's there, you're gonna.
Yeah.
So you're not restricting.
No.
Is that what you're asking, that she wasn't restricting the search to just one?
yeah, I was just curious of like, if your search was just like based on like, okay, first level parasitism, or you're just going against all of like NCBI, just single.
All of NCBI, yeah. So one thing that would matter is there going to be a difference in coverage, right? So if the hyperparasitoids have a different GC content or a different coverage, then it'll be really easy to see them, that there's at least something different happening there, right?
Because like Dendroceras have an unusually high GC content.
Yeah.
Those are the other ones like acids.
Yeah, it would be, I just wish we had, I feel like, I know that like 10 years down the future, we're gonna have excellent NCBI reference genomes, and we'll be able to like really look for that kind of stuff. But I'm sure right now I could blast my sequences against the whatever thing you said.
There I should be in.
Yeah. I'll check that out. That's a great idea.
Yeah, great thought. A lot to think about. The quantification question that Paul came up with was exactly on my mind, except from a slightly different perspective, in that as a parasitoid hunting an aphid in a soybean field, even though they're not moving around a lot, that's quite a task for something that's maybe about the same size as an aphid and struggling through the air column, et cetera. So there's a lot of incentive on finding the host. But if hosts are hybridizing and they're all kinds of goofy plants, it'd be interesting to the parasitoid has got to like figure that out. They want to exploit that resource. They're gonna have to figure that out. So it'd be interesting to look at how prolificous or how much interchange there is between different lineages of parasitoids as well. Like, what's their story? Because I bet there is one, and you're already capturing data as bycatch.
Yeah, I really think I think this is a really awesome idea. And this is like one of the awesome benefits of shotgun sequencing is like the bycatch, right? I do know, I talked to some of the people at the Wellcome Sanger Institute that is working on doing a lot of these, trying to sequence a whole bunch of genomes or whatever. And there was someone there who was actually working on, like, she would take all of the plant data that they had and somehow like a rather large percentage of the plant genomes that she downloaded actually had like aphid DNA in it. And then I think it also, there was some that actually had like Buchnera DNA in it, which I don't really understand how. Yeah, I don't know, but because there was plant data available, she was able to pull out the other DNA data from there. So I think we're really close. And to be honest, like I knew about our one sequence that had parasitoids in it because of it came out in the wrong spot in the tree, right? But actually, as I was running blob tools, I came to a second parasitoid that just randomly, like I hadn't even picked it up. I wouldn't have picked it up if I didn't use blob tools because it was in the right spot in the tree. But like, so if that's 2 out of about 30, then I feel like we're gonna be able to find some good bycatch to be able to start getting to that question. But yeah, the prolificousness of the parasitoids is
and the other thing that's odd about the system, going back to the ants, is you often have an invasive ant on a crop plant making a pet out of an invasive aphid that it's probably never encountered.
Yeah.
Like there's definitely elements happening here that are not, it's like we're chasing the wrong character system. They're seeing the world in a way we're not expecting. And we're saying, oh, isn't that weird? When they're just like doing their thing.
Right, yeah.
We're kind of missing the boat on some of these things.
Yeah.
So, and this is a really neat way to unlock that.
Yeah. Yeah, I think so. True. Yeah.
That's a really great talk. I would like to ask you, because these are pest insects, right? And we know that most of the hybridizations like don't have a good fitness for the hybrids, right? And just fewer hybrids can have like a better fitness, and these are like a pest and all these things. Like, have you done like any tests to see if the hybrid generations do back crosses, or they just, like the first generation of hybrids just die and they don't continue reproducing? Like, I know that there are some tests that we can run to see that. Like, have you done it, or have you? Or
I haven't done those tests yet, but I think that would be really interesting to see. Like, is it a hybrid event that becomes a new lineage, or is it a hybrid event that back crosses? I think some of the tests do tell you which one it is, but I think
dead end hunter, it's
Yeah.
the most frequent that
Yeah.
like happens in nature, but also aphids
Yeah.
can create surprises.
Yeah. Well, I think the important thing to remember with invasive species too is I think we talk about invasive species as being so successful and so wonderful, but they're actually like most invasions fail and we only see the ones that succeed because they didn't fail. So, okay, ready for some songs? Yeah.
We're trying to figure out how to get the camera for the show.
You can't.
You could try like
That's all right.
That one.
you try the pinning option where you can like pin it.
Yeah, that's it.
Yeah.
Yeah.
That's just Russ.
No, that's just me in my back.
Oh.
Okay.
No.
Allison.
I don't think that's.
You can trade back.
You get there.
It's like a theater in the round.
It was both from.
Yeah, you gotta like rotate.
We're in the round.
Okay, so
Oh, I had one more question.
oh yeah.
So I know I'm after a classification change laws. I learned it, but are all hemiptera now called true bugs? I thought it was still just like Chiroptera.
I think it's just
It's like Stenorica got moved into hemiptera.
yeah.
True bugs and allies. We call them unnaturalists. It's true bugs and allies.
Yeah, I think the
Oh, they're
Stenorica
good
is
bugs.
not-
Okay, so it's not a true bug, it's just the.
I feel like people outside of Heteroptera just kind of refer to Hemiptera as true bugs, but Heteropteras are like, no.
Not a true bug. All right, I'll delete that off my slide next time.
I don't know what all the
Aphids.
friends are.
Plant lice.
Allies.
Like all the
Yeah.
sucking insects.
All right.
I actually had another question.
Oh yeah.
Bugs and friends.
Very basic. I was just, yes. You mentioned how they five-year officially and then they have like one generation.
Like
sexual
fun, yeah,
males. So there must be very little genetic diversity with the constant asexual production. So does the hybridization kind of negate that if there is such?
This is one thing I would love to look into. I would love to figure out, because evolution isn't happening at the same pace if they're only having one reproduction.
Because you're looking at more
Yeah.
between species. I was thinking more within one species, how much genetic diversity can be introduced through the hybridization.
Yeah, well,
That could benefit the species in
Yeah.
long-lasting and protection against disease.
I mean, it might be that they need hybridization with different species because they only have one chance at reproduction per year. So that's a really important thing to think about. Yeah. All right, this first song is called Superorganism. I wrote about termites. I actually could put the lyrics up for people because I don't know if they'll be able to hear.
Sing along, like Carrie.
We'll sing along in the last verse. So here we go.
Termite.
First, actually.
Okay. So
Super. Let's get rid of it.
Can Zoom see it or no? Probably not.
I stopped sharing to make the thing come back up, so you can share again.
Sure.
Oh crap! I did play from beginning. Got Zoom. Share screen. I've got. Okay. All right, here we go. You're a superorganism, part of a team. Superorganism, you know what I mean. There's queens and kings and workers and soldiers, building up a termite mound the size of a boulder. You do your part and we'll do the rest. Superorganisms, we are the best. Oh, you might be an immature, hatch from some eggs. Like other insects, you have 6 legs. When you grow up, you're not sure what you'll be, but anyway it goes, you will be part of a team. We are a superorganism, we're part of a team. Superorganism, you know what I mean. There's queens and kings and workers and soldiers, building up a termite mound the size of a boulder. You do your part and we'll do the rest. Superorganism, we are the best. Oh, you might be a worker who tends to clean with care. foraging for food that you can bring back to share. Tidying and cleaning up the termite mound. Workers are the termites that you like to have around. Oh, and ants, birds, or reptiles come in for an attack. Soldiers are the termites that stand to fight them back. Oversized heads and mandibles that chomp, signaling there's danger with a loud and solid thump. We are a superorganism, part of a team. Superorganism, you know what I mean. There's wings and gills and percent soldiers building up the termite mound size of the boulder. You do your part, we'll do the rest. Superorganisms, we are the best. Oh, if you are an alien, you have it pretty good. Other termite workers bring you sacks of termite food. Your only job is growing out your wings. One day you'll fly up to become the kings and queens. Oh, if you are a queen or king, you're lucky. Least of all the other termite workers tend your beck and call. They bring you food and they lift your mood. Well, you just keep on making termite brood. I sing with me. We are a superorganism. You're part of the team. Superorganism. You know what I mean. There's wings and kings and workers and soldiers building up the termite mound size of the boulder. You do your part and we'll do the rest. Superorganisms. We are the best.
I have a music question.
I have heard my question.
There's no questions in the song.
Yeah.
Out of order. Never talk about.
All right. Oh, it's fine. I just wanted to know if you were. I'm gonna ask it anyways. I wanted to know, are you gonna like release these on like Spotify or SoundCloud? Do you have like?
I'm gonna make a book, and they're gonna sell the book in the system. And they're gonna have a YouTube channel or whatever. But I'm gonna end with my better song, but this is one. This is the one that I'm in the process of writing. I'm actually doing outreach event at the Smithsonian. And they do like play date family days where I'm gonna sing these songs, and the day is like a pollinator day. So this is a special song that's written just for pollinators. And yeah, it doesn't have all the verses yet, but and it's supposed to be played on an electric guitar. It's a little fuzzy, but this one is this is what we got today. Pollinator, pollinator, buzz, buzz, buzz. Pollinator, pollinator, just because. Plants gotta grow, gotta make some seeds. So pollinator, pollinator, come on, please. Oh yeah, we love pollinators. Yeah, they're good for everyone. Oh yeah, we love pollinators. Buzz, 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 buzz. Then we got
Shades of Seven.
Oh, you probably heard of a honeybee. She does a little dance and she makes honey. But you should know that she's not the only one. Many other pollinators get the job done. And then there's gonna be like a verse about like beetles and flies and bats, and then we end up with, if you have some space to plant some native plants, we believe so the baby pollinators stand a chance. Cut back on the insecticides and let's help the pollinators live good lives. So pollinator, pollinator, buzz, buzz, buzz. Pollinator, pollinator, just because. Plants gotta grow, gotta make some seeds. So pollinator, pollinator, come on, please. Oh yeah, we love pollinators. Yeah, they're good for everyone. Oh yeah, we love pollinators. Buzz, 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 buzz. Alright, I've got one more. If you guys, this one's like a guessing song. So
It's interactive?
this is interactive. This one is interactive. So
insects and their problems.
Yeah. I also only know like 3 chords on the guitar, so. Okay.
There's no word enough for this one.
I'm an entomologist, I look at bugs for fun. I'm an entomologist, out under the sun. If you see an insect, call me. I'm an entomologist, I'll be right there. I don't think I got the right chords. Close enough. All right. Thank you. Oh, I'm singing the other song. Okay. If you see an insect with elytra on its back, crawling all around the ground, searching for a snack, biting, chewing mandibles that might be painful. It's probably okay. If you see an insect with just one set of wings, flying all around your house, crashing into screens, stubby short antenna and big round eyes. It's probably Alright.
I like this song.
Circling the light, spreading out its wings as it is landing from a flight. Feathery antenna and it's furry like a sloth. It's probably a
Well, we're really good.
If you see
100% so far.
We're in the right field, guys.
if you see some tiny bugs that suck the sap from plants, making balls of honeydew that get slurped up by the ants, little clones can reproduce before they get it. It's probably
Not a true bug.
Ally.
This one is a little bit of a stretch. An insect hovering near a pond, giant eyes to help them see the bugs that they prey on. They move their forewings separately, which helps them zag and zag in the sky. It's probably
Or damsel.
This one's also a little rough. With giant jumping legs, long antenna help them sense the danger near their head. You can often hear its chirp coming from the thickest.
Solid.
It's probably Alright, last one. If you see some insects walking in a line, following the pheromone trails with the food they find, you better not get too close or they might throw up your pants. It's probably some. Very good. Thanks for listening.
So cute.
First ever songs that are.
I would like to make a verse for some really cryptic thing no one's ever heard.
Excellent.
If you have a verse, let me
It
know.
might be a scorpion fly.
Is there any further business? That was wonderful.
Yeah, that was adorable.
No objection, the meeting is adjourned.
You gotta get some long genus.
Excellent.
Incredibly long Latin genus for your last one.
Yeah, and
Here, I
like-
didn't get this one.
Yours
Everyone's
right
feeling
up.
real confident.